So today, I'm going to actually start by trying to kind of subliminally share some things with you, and then also go on to what clinically we're trying to do here. Okay, so I'm going to be talking about a new, basically, paradigm approach, bipolar androgen therapy. But to give you this, I have to kind of give you the background of where this came from. Now, androgen regulates the growth and survival of both normal and malignant cells, but it does it in a vastly different way. This is really a fundamental thing, that in normal prostate is an indirect paracrine interaction between growth factors being produced by the stromal cells under androgen stimulation. The epithelial cells are essentially programmed by those growth factors. Androgen receptor does not drive proliferation of any epithelial cell that I know of in the human body, other than cancer. So even in spermatogenesis, there's no part of spermatogenesis in which one of the sperm cells expresses AR. That's all a paracrine interaction by supportive cells. So normally the androgen regulation is paracrine. In cancer, it becomes autocrine, and we'll see there's another very important difference. In fact, the reason that the prostate doesn't overgrow in the presence of continuous androgen is because the luminal cells engage their androgen receptor not simply as a transcription factor to make PSA, it actually binds to various cofactors to turn off the production of MYC. So the androgen receptor's function in luminal cells is a growth suppressor, not an oncogene. And yet, we know in their publications, we can look these up, what have I just done? Just made it go to that. Excuse me. However, during prostatic carcinogenesis, the androgen receptor is actually gained, not as a tumor suppressor, but as an oncogene. And for many years, we've known that it suppresses, drives the growth. We can study it in tissue culture. And what's happened is we've tended to think that it's acted as simply as a transcription factor, making more growth factors in this basically autocrine type pathway, where the androgen receptor definitely is a transcription factor. It definitely is making enzymes like PSA and HK2 and other growth and survival factors. But that isn't the only thing that it does as part of its oncogenic activity. It also is involved in DNA replication. It's important as a DNA licensing factor. And let me go over what DNA licensing is. Cells have an interesting problem. They have to make their DNA able to be essentially copied during S phase, but they want to only replicate DNA once, and only once per cycle. So they have to figure out a way of regulating when and how much times DNA can be copied, and that's called licensing. So normally what happens is that you have a cell that's been recruited to start the essentially G1 part of the cell cycle. At origins of replication, these are specific sites between 20 and 30,000 sites per cell. They're not a sequence-specific site. Every lineage has different origins of replication. That's a very interesting question of how are origins of replication defined. But we know that what happens is when cells enter cycle in G1, they can license their DNA by having essentially a complex of origin of replication proteins bind to these sites. These then recruit other partners, the CDC6, CT1. These are needed to pull in what are called the MCM proteins, which are the mini chromosome maintenance proteins. This is needed to allow the chromatin to be marked so it can actually undergo replication in S phase. Now, what's happened in the androgen receptor story is in prostate cancer, unlike other cell types, these origins of replications are being formed in the early G1 stage, but it isn't just those origins of replication proteins that are at these origins of replication. The androgen receptor occupied by androgen actually binds there. It then actually is involved in the complex that is involved in this licensing that occurs in G1. As the cells progress from G1 into S, they have the interesting situation that their DNA is now licensed and replication can occur, but in order to prevent relicensing coming around, they have to degrade a portion of those licensing factors. Androgen receptor is not one of the factors that gets released during this integration into S phase. It stays along with these origins of replication complex. And it actually is maintained all during S phase as part of the origins of replication and DNA replication. However, during the G2M phase, it has to be degraded along with the origins of replication program to allow licensing to occur in the next cycle. So every cycle, you essentially license in early G1, you go through S, you restrict the ability to relicense by keeping the complex bound, and then you degrade the complex, with proteases during mitosis. And so normally this is going on fine, and androgen receptor is helping this. But the interesting thing is that this requires a critical amount of androgen receptor, and that amount of androgen receptor produces a liability if it becomes too high. So during castration, what you're having is not only, so here's a typical example of a patient whose PSA is rising, given androgen ablation. That acts basically as a culture shock. It essentially is stopping the production of growth factors, survival factors, and the cells aren't, they're either undergoing apoptotic cell death or they're not licensing their DNA. That's fine. So you get a regressive response. There's a period of time when there's an adaptation. We don't really understand what drives this, but essentially, almost universally, in a very large subset of patients, PSA will start rising again, and this is usually associated with an upregulation of the androgen receptor protein itself. Now, we know that the androgen receptor protein is involved in autoregulation. There's a whole microRNA network for regulating mRNA for androgen receptor. So normally, you've got a unit amount of it that's plenty for replication to make the growth factors. Everything's fine. When you have a low level of androgen, it upregulates the amount of androgen receptor so that it can generate the same signal with a much lower amount of ligand. However, and it can happen that way because either through amplification, a variety of mutations, a variety of things. But this is really something that I think a lot of people haven't kind of realized how much amplification we're getting here. So if you look in this panel, if I can get this pointer, if you look in where it says A, where it says normal prostate, that's the luminal cell that has 1 unit of androgen receptor. If you look at local prostate cancer before androgen manipulation, it has about 3-fold. So it's about a 3-fold increase going from a normal suppressor to an oncogene. That's the magnitude of increase. When you look at the cell lines that we grow in culture, they're all derived from castrate-resistant patients, and they have an incredibly high amount of androgen receptor. It's not trivially increased, it's a major increase. If you look, this also occurs when you're looking at mRNA expression profiling of samples from normal to malignant to metastatic cancer resistant, castrate resistance. There's also this amplification of the androgen receptor mRNA. And the question is, can you take therapeutic advantage of this amplification of the androgen receptor? And the answer is yes. Because what happens, go back. The answer is yes. And we heard about, when Karen was talking about this idea of synthetic lethality, this is basically a type of induced synthetic lethality, because as these CPR cells amplify and upregulate their androgen receptor, they have the problem that if you give back a high dose amount of androgen, a pharmacologic amount of androgen, not a physiological amount, but a very large amount, you can eventually stabilize the androgen receptor during mitosis so it can't be degraded sufficiently to allow relicensing. So what happens here is that you can see in this middle product, immunoblot for androgen receptor, those are cells grown with their happy amount of androgen. If you restrict them, they adapt, they upregulate their androgen receptor 3 or 4-fold. Okay, now what happens if you take those cells and expose them to R1881? They don't just stop growing. They try to go through S phase, but because they haven't degraded their androgen receptor sufficiently in the last cycle of mitosis, they can't relicense in the next phase. So they get into early S phase and they actually die. So you can take advantage of this then. Now, the interesting thing about that is you'll kill a certain number of cells, okay? But eventually what happens is they're survivors because the cells auto-regulate and they down-regulate. They go from 3.5 back to about 1.6. So you have a unique window of opportunity. When you've used a castrate environment, they'll upregulate the androgen receptor. If you give them a high dose of androgen, you can kill a certain number of cells. However, there's a built-in mechanism essentially of downregulating the androgen receptor in the survivors, but if they downregulate and they now are exposed to a low level of androgen, they've now got another synthetic lethality because now they can't survive in the low castrate environment. So the question is, we've shown in multiple models that if you cycle rapidly between high pharmacological amounts of androgen to very low castrate levels, and you do it rapidly enough, you can essentially catch the cells out of phase. They're trying to adapt, but they're always not quite catching up. So you can get cell kills during this. So the question is, can you actually do this in people? And so Sam Denmead is the clinician that has driven this about using testosterone not as a hormone. We're using testosterone here as a drug. It's essentially trapping the androgen receptor in mitosis so that it's not fully degraded. So this is pharmacal amounts of androgen. And so what Sam developed was a really nice protocol. Actually, do I not have that picture? So what he came up with, you can imagine trying to tell urologists that you're going to give back androgen to a castrate-resistant patient. So initially, to get any clinical trial started, he realized that if he combined it with basically using other drugs like topoisomerase inhibitors, which had shown to be effective with essentially when you have double-stranded breaks, you could at least have the idea of having some chemotherapy while you were doing this at the same time. So in the initial trial, there was a combination of injecting men with essentially topoisomerase and an intramuscular injection with 400 mg of testosterone and cypionate, a very short-acting androgen. It'll actually make, that's why I guess I left the slide out, but it's where they're showing that the levels of androgen go up to about 10 times physiologic. So we're not in physiological range, we're in pharmacological range, but it's only up there for 2 weeks. So in a patient that has been maintained for, these patients were actually 45, mean times 45 months on androgen ablation therapy, primary or secondary. So these were castrate-resistant patients in the first study. And what Sam was doing was injecting every month and producing a spike of testosterone that would raise it to pharmacologic amounts, followed by essentially a crash down to castrate levels because you've got LHRH analog. And these are the first 15 patients that he had. These were patients, half of them were essentially low metastatic burden. They were rising PSA. They were asymptomatic. This was just to kind of to prove that he could do it safely. And in fact, he got significant PSA responses. He actually got objective responses in these patients too. So it wasn't just simply a change in PSA. You can look at the actual, the most recent publication for all the details on this, but it was quite interesting. Soft tissue lesions actually got smaller. So this was not just a PSA biomarker change. And he saw this in multiple patients. So about more than 40 to 50% of that first cohort of patients actually had objective responses. So then he actually started thinking about the idea of, well, okay, we'll do this in men that actually have a much more metastatic disease that have been actually progressing on either enzalutamide or abiraterone, with the idea of, could you actually not only get this response, but could you also resensitize them to essentially androgen, these other forms of androgen, because the androgen receptor levels are going basically up and down, and you're resensitizing them. So in this trial, which he called the RESTORE trial, which hasn't been published yet, the same situation, LHRH analog continuously. These are patients that are castrate resistant. They've either been on abiraterone or enzalutamide. After they progress on that, they're placed on this therapy every month. Now in this treatment, there's no topoisomerase inhibitor. He became brave enough, I forgot to point out in that last group of patients, many of those patients went on for almost a year of this cycle, and they only got topoisomerase for the first 3 cycles. After that, they were only on testosterone. In this trial, they were only given the injection of testosterone, so it's a high up with testosterone and a thing coming down. And again, he's got the waterfall plots. And the interesting thing about this situation was when the patients progressed and their PSA went up, they could be put back on either enzalutamide or abiraterone. And what he actually found, well, I should say that he also, again, in this patient, these groups of patients, these were some liver mets. He got a series of patients that actually responded quite well to the soft tissue. But here's an example of where these patients have been resensitized then to be crossover with the endo after they progress on the BAT therapy. They can be responding to abiraterone or enzalutamide. So he's got a bigger trial, which is a multi-cooperative trial that's going to be done through the DOD, which he calls a transformer trial. And this involves the same type of situation. Patients that have been on LHRH analog, continuous situations, they're going to be on abiraterone for a period of time. They're either going to, when they progress, they're going to be either put on enzalutamide or BAT, and they can be crossed over to see if they can be resensitized. And so this is a big DOD trial that Sam's running, and there are a number of sites that are involved. So this is actually, I think it's 150 patients in each arm. But this is going to be a very useful trial because, again, these are a very specific type of patient. This is not for early prostate cancer. These are for patients that have actually gone through all of the hormonal manipulations where a subset of patients have been selected for the basis of having androgen receptors still be a main driver, but be autoregulatory, so that you go through these cycles of essentially synthetic lethality. So the take-home points are that this bipolar androgen therapy, where you're cycling between very high levels of testosterone and then within 2 weeks going down to castrate levels and repeat it. So you're not giving this. This is not like intermittent androgen blockade where you've lowered the androgen and you're waiting for it to come back up. You're going through every time these cycles. And so first of all, these cycles have turned out to be safely given to men that are asymptomatic. You get objective responses. It does resensitize. And one of the things that Sam has really been impressed by is also the quality of life issues in these men. And so I look around, there's a series of people that are actually involved with this as part of the clinical team that are here, talking. Michael Schweitzer was one of the main fellows that drove this originally, but as you've heard, we have a very active group and a cooperative group at Hopkins. So this, we think, is going to be a very useful approach. It is not for everybody. And I want to make sure people understand this. This is not the idea that you can give back androgen to patients chronically. The issue here is to do it episodically so that you're taking advantage of the fact that you're not letting the cells adapt. And I think it actually is a paradigm for many other types of therapy, where we've essentially given a drug and we think we can give it forever. We wait and let the cells essentially develop resistance, and then we'll start figuring out how to change. It's in those rapid changes of the microenvironment that the cancers are most susceptible. So I think that there's going to be other types of therapy like this, where you want to go from something that's where you're taking advantage of the fact that for a cell to be resistant to a therapy, it becomes sensitive to another therapy, and that's called an induced synthetic lethality. So I think this is going to be a very interesting series of events of using this. So I will stop there and take any questions.
So
I can almost see you.
No, it's fine. So I'm actually a huge supporter of this concept, and we tried some of this a number of years ago. We're participating in this trial. But one of the fundamental questions that I have is, this is really based on the cancer adapting to various, androgen environments.
Mm-hmm.
But from some of these genomic studies in late disease, there also appears to be clonal selection of variants that, are not just adapted. These are, genetically altered.
Oh, no, I agree.
And at what point in this natural history is, adaptation changing? Are we changing from adaptation to clonal selection? And therefore, at what point in the clinical history should we be doing this kind of treatment?
Yeah, my earlier life, I spent a lot of time on clone selection, and I worked with, it's an interesting example, I worked with rat tumors when we did not have human xenografts. And what would happen, we started off with cancers that had androgen receptor and were highly responsive to hormonal therapy, like the human situation. But the rodent, the rat in particular, was the problem that very quickly it would develop androgen resistance by not expressing the androgen receptor. It would turn it off completely. And so in my career, I decided to turn right. I was going to say, okay, I'm never going to work on androgen receptor again, because what's killing men are essentially AR negative tumors, I thought. And then we had the more recent revolution of looking at really human tissues and human xenografts, and lo and behold, they're different than rats. So let me repeat that. Human prostate cancer is different. Not all. No. PC3, D145. And we even have autopsy studies where we have men that we get samples from, and we've established xenografts from them where they are completely AR negative right from the get-go. So let me back up for a second. I've lived long enough to never say that prostate cancers will all do this, okay? There's going to be a subset of people. But what I was really excited about this was the idea that these are patients that are basically, they're kind of selected on the biology that they have. Many of these people have responded, and they've been able to go long enough. We're talking not people that have been on androgen ablation initially for 6 months, but many of these people are on for 4 or 5 years, okay? So the question is, do I think that there'll be patients that will have who will have gone very rapidly through androgen ablation, and you give this to them, will they do anything? No, because I think you're right. Those are the ones that will have figured out how to not use the androgen receptor at all. None of this will be helpful for that. So this is for a very specific subset of patients. The interesting part of it, though, is it's kind of the patients that nobody wants. It's the ones that have failed everything. So the good news is that you're allowing I shouldn't say good news, that the issue is that you've allowed enough time for the selection. So I do think that one of the things that will happen with this is that you do have to, it would be nice to have a biopsy ahead of time, but I do think that there is a real drive for human prostate cancers to remain at least some level of AR positivity. So I'm agreeing with you. I don't know what the percentages are, but at least the other thing about this is that, like I said, that last comment about that Sam was seeing, Sam is a rational human being. And initially, working with these patients, they get very enthusiastic about this. These are people who have not really enjoyed life. And the idea of you're telling you we're going to give them back androgen initially, you have to kind of convince them of that. But after they start getting the injections, they come back early. They want the injections. So compliance has not been a big problem here.
Great talk, John. It's always really exciting to think about. So 2 related questions. One, do you know what the hormonal profile actually is in patients who've been, intratumor hormone profile in patients who've been on bad therapy? And related to that, since you're using an aromatizable hormone, is estrogen the real active agent here?
Well, again, maybe I didn't express that correctly. Estrogens are not stabilizing the androgen receptor.
But there's ER. There's ER in the tumor. There's ER in the tumor microenvironment. So
right, that could be, and you're bringing up another interesting point about how high dose DES, and many people who are no longer with us used to think that was a great drug, even in people who have been surgically castrated, that it has effects on microtubules and others. So I don't know that, and you are correct that testosterone is aromatizable, but this level of hormone is drug-like. So it's up really high fast and it comes down really low. Sam is actually measuring the estradiol levels though. In the first study he didn't, but in all the rest of them he's profiling the hormonal profile on them for questions like that to see if there's some unexpectedly high amount of estrogen. But he hasn't had, the other kind of sequelae, what you might think of be high estrogen side effects. So he hadn't.
Well, maybe, and I'm not sure that the serum are going to tell you the story. I think
Right.
It's in that scenario you're really going to have to profile the intratumor hormone levels to get to the answer of how this is actually working.
I agree.